Welcome to FOOD2000. My name is Maggie and I'll be showing you how to use the equipment. Today we will look at Practical Module 1, Rapid Viscoanalysis, which we call RVA. And here it is. Let's start with a bit of theory. The RVA measures the viscosity of a food or ingredient and provides information about the temperature that cooking begins, the swelling of starch granules as cooking proceeds, and the retrogradation or hardening of starch as it cools. The RVA can give us this information for flour-based foods, dairy, fruit and vegetables, and all sorts of processed food. We prepare samples by grinding our food into powder or flour and then simply mix that with water. This creates a slurry. We then cook the slurry and cool it. Now let's look at how the viscosity curve forms. The RVA heats from 50 degrees Celsius to 95, holds at 95, and then cools to 50. The sample is stirred constantly and resistance to stirring is recorded as viscosity. The inflection points of the viscosity curve are consistent across samples because they are a function of heating and cooling, but different samples have different values at those inflection points, giving specific information about the sample. Now we are ready to use the RVA. First, we need to put on our personal protective equipment. We need our safety glasses, and you will need a lab coat. You will be given powder of your food, and then using a balance, carefully weigh 25 grams of water and put this into an RVA canister. Then weigh 3 grams of powder and add this to the canister. Stir the contents of the canister with a stirrer. Place the canister into the RVA, select the correct program, and then press Run. The RVA will take 12 to 15 minutes and end automatically. When the run is finished, export your file as CSV. Let's talk about analysing the data. We will then plot the file of viscosity against time, which must be converted from seconds to minutes. We need to find the values for peak viscosity, trough viscosity, and final viscosity. We also derive some parameters and look at setback, breakdown, and hot paste viscosity. So plot your curves, find the value of each parameter and derived parameter, compare the curves and explain why there are differences between your different food samples. Enjoy and see you next time!